Cytogenetics is the study of chromosome structure and the formation underneath the microscope, and it is a common technique used to observe cells undergoing mitosis, as the structure of the chromosomes is different depending on what phase mitosis the cell is in. From a sample of tissue in which DNA has been stained by a colored dye that binds DNA, cytogeneticists can count the number of cells in interphase versus mitosis, and they can even count the number of cells in the various phases of mitosis. From this, cytogeneticists can actually calculate the mitotic index, which is the number of cells in the mitosis in a given sample divided by the total number of cells in the sample. Mitosis is essentially the process that cell division in somatic cells in organisms. One parent cell will form two genetically identical daughter cells, which is identical to the parent cell. The time period between subsequent mitosis events or cell divisions is called interphase. During this time, chromosomes is not visible under a light microscope. Instead, DNA is present in the form of chromatin. During interphase, the DNA and the organelles inside of a cell replicate. The four stages of mitosis itself are prophase, metaphase, anaphase, and telophase. During prophase, chromosomes become more condensed and is visible under the light microscope. Specifically, each chromosome consists of two chromatids, which was formed from DNA replication and interphase. At the same time, there is an organelle called the centriole, which is replicated when each moves to opposite poles of the cell. The nuclear membrane is intact until the end of prophase. During metaphase, the mitotic spindles will grow from each of the centrioles and attach to the kinetic core, which is a protein complex that binds to the centromere. And the centromere is actually the center of the intersection of two chromatids in the chromosomes. Then the chromosomes all line up at the equator of the cell, as you can see in the diagram. During the next stage called anaphase, the chromosomes move away from each other, move away from the equator, and move towards opposite poles of the cell where each of the centrioles is located. Specifically, each chromatid in the chromatid pair that makes up the chromosome is pulled to an opposite pole of the cell from the other chromatid, and this is done by the microtubule spindle fibers shortening. The last phase of mitosis is telophase, and during telophase, the sister chromatids at the end of each pole begin to unwind, exiting their condensed state. They no longer are visible underneath the microscope, and the spindle fibers in the cell also break down and the spindle disappears. A nuclear membrane forms around each set of sister chromatids at the pole's ends. After telophase, an event called cytokinesis takes place, where the cell, which now contains two nuclei, split into two daughter cells, each one containing one nucleus. The final result, as mentioned earlier, is one parent cell giving rise to two daughter cells, each genetically identical to one another, as well as the parent cell and containing the same amount of DNA content.